All right. Thanks, everybody. This is my actual preferred time to give a talk, so I missed the because I have a and if y'all fall asleep during my presentation, it's because you just had lunch, so it's not my presentation. But yeah, I'm really honored to have the ability to talk to you all today. We're a new member of the KS research community, but already I've been learning what an awesome community it is and learning a lot about the cool research that's been going on in it. So yeah, thank you for the introduction. Yeah, our group works on kind of new gene therapy technologies, very early preclinical development. So my goal for this presentation is to explain a little bit about what we've been working on. Hopefully you understand a little bit how it works and share some preclinical results. So yeah, I'm in the lab of Professor Brian Dickinson, and can get started. You just have to like close it out and open it back up.
Sure, it froze earlier and then it just started working.
Okay.
There we go. Okay. It's not working. Oh, it's working.
Okay, great.
Yeah, it's just a little bit slow.
Okay. All right, so I know it obviously doesn't need a lot of external validation for why we need drugs to go after neurohaploinsufficiencies, but I like to start with this little graph just to kind of contextualize what we're going to be doing and what our... So here at the bottom, we have examples of what a healthy genotype would look like, where you have two healthy alleles. And then on the y-axis, we have protein expression, where we have some level of protein that's in a healthy level for normal physiological function. We have genotypes which cause abnormal protein expression, either like through a duplication, so we have too much protein expression. We have a lot of drugs to either decrease protein expression or inhibit that protein to correct that state. But in cases where, like haploinsufficiency, where one gene has a mutation that leads it to be non-functional, this is a much bigger problem. And it's also important to contextualize that we want to increase protein back to the healthy protein levels, but there are many genes that are actually sensitive to both gene under and overdosage, so we always need to be cognizant of that. And something that's really amazed me getting into this space is there have been some bioinformatic analyses of early like clinical genetic data that have suggested that there are many, potentially thousands of genes sensitive to haploinsufficiency, which may not also all obviously need to be treated, but it's just like we just need to have better tools to meet a potentially growing need as genetic testing moves further into the clinic. And when you actually look at where these are clustered tissue-wise, the brain is heavily enriched, so things that are actually delivered to the brain. So if we think our end goal is to increase protein production, so if we think back to like Bio 101, how do we get to protein? Well, it's the central dogma. So we go from DNA that's transcribed into mRNA, and then the mRNA is translated into protein. So if our goal is to make more protein, theoretically, we can along this process. And as you're going to definitely hear in the there's a lot of really cool ways that we can tackle this on the DNA level, where we're trying to get more mRNA to be transcribed, which then leads to more protein. And that trend kind of uses as we move along in the dogma, where if our goal is to get more mRNA, there are a lot of cool emerging technologies to do that. But if we get near the end, where we're actually the last step, where we're turning over the mRNA to be translated to protein, there's a lot less explored space here. So our lab, working on RNA targeting technologies, are really interested to look at that. And of course, one technology is not going to be able to solve haploinsufficiency-related diseases broadly. We're going to need a lot of different types of approaches, which some will be fit for some genes more than others. But we're explore this strategy of actually trying to intervene at this last step to increase translation of an mRNA into a protein for some advantage, or that has some advantages that I'll go over in the next slides. But before I get to that, I want to talk a little bit about the system that inspired us to go down this path. And it actually came from across the hall in the lab of Professor Chuan He, who his group works on RNA modification and RNA binding proteins that read those modifications. And they described a system where mRNA can become methylated within the cell. And then that read out by what we call reader proteins. And these proteins are really cool. They're kind of like bifunctional molecules in a way, where we have one domain of this protein that binds this methylation event, and then the other domain which recruits other proteins to do some sort of chemistry at this mRNA when it's in close proximity. And for the case of this protein Y1, that is actually an increase. So we were thinking, well, we don't want to target every protein that is every mRNA that's methylated within the cell. Can we replace this RNA binding domain with so that we can actually bind an mRNA of interest? And a previous graduate student in the lab, Simone, did this, where she took the here of Y1 and expressed it as a fusion to a CRISPR-Cas molecule. So if you're familiar with CRISPR-Cas molecules, they're a protein that actually binds a separate RNA called a guide RNA. And this guide RNA can be programmed to be complementary to a nucleic acid of interest. And in this case, we used CRISPR-Cas13, which natively targets RNA molecule that we're targeting. And we used a catalytically dead version of this. So we're not degrading the mRNA, we're just doing binding. And so this fusion with the effector translation activating domain of Y1 with the RNA targeting Cas13, when targeted to a mRNA that encodes firefly luciferase, we saw that if we measure the enzymatic output of firefly luciferase, which is light, compared to a guide RNA that does not bind the firefly mRNA, we see that we have an increase in luciferase expression with the on-target guide. So this just validates the idea that we can build programmable translational activators that target mRNA and a specific mRNA of interest. But if we actually wanted to use this in a therapeutic context, there are a lot of limitations. One, Cas13 is huge. It's too big to package in FDA-approved AAV vectors itself, so this was not going to work for that. As well as they're of bacterial origin, so they have immunogenicity concerns from the extended expression that you would need to do for an RNA targeting modality. So our lab developed a separate platform called CERTS, or CRISPR-Cas-inspired RNA targeting system. So this recapitulates the functionality of the Cas13 molecule, but using engineered human proteins. So for example, we have a RNA hairpin binding protein shown here in blue in the middle. That's going to bind our guide RNA, similar to how. And then we have another single-stranded RNA binding protein that's expressed as a fusion to this blue, the guide RNA from degradation within the cell. And then to these two proteins, we also one effector domain that I showed previously, and we can see the same result that we got with the Cas13. And this is significant because this version is over 50% smaller than the Cas13 modality, so it easily fits in viral vectors for delivery, and it's engineered from human proteins, potentially mitigating some immunogenicity concerns. But this is where I really entered in the project, where this was cool results on reporters, but actually translating this to targeting disease-relevant transcripts was going to be a whole project onto its own. But just to kind of recap, the idea here, if we wanted to target a haploinsufficient mRNA with CERTS, so we should have a pool of 50% of the healthy mRNA that we'd want to program our guide RNA to bind to. And upon binding to the disease-relevant mRNA, our CERTs, they're going to recruit proteins in the cell called EIFs, which are eukaryotic initiation factors. So this primes the mRNA to recruit the ribosome to start translation. And this is actually the rate-limiting step for virtually almost all mRNA in the cell. So this is really like the step that the CERTs actually helps with. And then once the mRNA is primed, ribosomes will land on the start codon and start making the protein of interest and increase target expression. And something that we're really excited about here is a cool, I guess, advantage of RNA targeting is that it has some cell type specificity baked in already. So if the target, if the cell that the CERTS is delivered to does the target, it will not express the mRNA, right? So every cell expresses the mRNA that it needs, and then the CERTS can't overexpress the target if it doesn't have the mRNA to work with. So cell type specificity, which is really cool. So to answer the question whether we could apply CERTS to a disease we have as a proof of concept, and I selected start developing certs around Dravet syndrome. So Dravet syndrome is a haploinsufficiency of a gene called SCN1A, which encodes a sodium ion channel NaV1.1. And it's a true haploinsufficiency in that 50% expression of this NaV1.1 protein leads to damage to the excitability balance in the brain and results in a childhood epilepsy. And the reason that we went after this is it has a very tractable mouse model to answer very clearly whether we can make phenotypic changes. And while there are a lot of really exciting clinical trials going on around Dravet syndrome, there's no approved genetic medicine as of this point, so it's still definitely worth working on. So to answer the question about whether certs can upregulate NaV1.1 expression, first we have to find a guide RNA that binds the SCN1A mRNA. And so to do this, we use the same luciferase reporter that I showed earlier, but now we attach the 3' untranslated region of the SCN1A mRNA to the firefly luciferase mRNA. So this allows us to express different guides that bind the SCN1A sequence and see whether they can upregulate the F-Luc signal. So it's just a higher throughput way to measure RNA binding. So here in this experiment, we transfect a vector that expresses the certs and a vector that expresses our reporter, and then we measure the firefly compared to a non-targeting guide. And this schematic up here shows a map of the 3' untranslated region, and these boxes are where the guide RNA that I designed land. So we have like the stop codon here, which begins the 3', and so we have guides that fall closer to the stop codon and also guides that fall near the end of the 3'. And using our certs Y1 that I showed, we found that guide 5 specifically was able to increase firefly luciferase expression, but when we actually went to target the endogenous SCN1A mRNA, this time in neuro2A cells, which are just an immortalized cell line that express a lot of neurotargets, by Western blot, we were able to see some modest activation, but it was pretty low. So this was exciting in that this strategy may be tractable to Dravet syndrome, but at the same time, we knew that we had to improve things. So I'll go over a little bit about what that preclinical improvement looked like, optimization. First, we looked for new effector domains using the same SCN1A dual luciferase assay that I talked about on the previous slide. And we chose effector domains to fuse to certs from the literature that people had described as potentially increasing translation. And long story short, when we did the validation on the endogenous mRNA, we found that one effector in particular, EIF4G, which is a master scaffolding protein for initiating translation, was able to upregulate NaV1.1 expression, also minorly, but we were excited that this protein works because it's very well studied, so it's a lot more tractable to engineer compared to Y1, so we moved forward with it. And the first step was we repeated the guide screen to see if the same guides that we try on Y1, if 4G has different preferences for where it likes to land in the transcript. We found that indeed it functioned both, at the end of the 3' did, but it also functioned right after the stop codon with this guide 1 also showing activity. On Western blot, that was confirmed. And then because this is a master regulator of translation that actually natively works in the 5' UTR, we reasoned that it should also be able to activate if we design a guide RNA that binds. And we found that was true. So the SCN1A endogenous mRNA that bind the 5' UTR here, and we could see some activation, about 50%. So this was cool because compared to Y1, which was natively a 3' UTR binder, it did not work in the 5' UTR. So now we at least have an effector that has greater generalizability as far as guide spots. But one big problem with EIF4G, no pun intended, is that it's very large. It has like 1,600 amino acids. So if we wanted to get this back down to be deliverable by viral vectors, we were going to have to do some optimization to its size. And so I did a truncation study. So up here is a map. Like I mentioned, it's a scaffolding protein, so it has domains that bind a lot of different partners to orchestrate initiating translation. And so we tried a couple of different truncations centered around this domain to bind a protein EIF3, which we hypothesized was most likely the most important contact because it's the protein that bridges the EIF4G to the ribosome. So these results showed that this truncation T3 was the most active by targeting the NAV1.1 mRNA by Western blot compared to a SERTs with on-target guide with green fluorescent protein as the effector, which doesn't increase translation, obviously. And this was really gratifying to see that EIF4A binding domain and the EIF3 binding domain worked together to give the best activity. One, because this actually validated some in vitro binding data that another lab had collected, that they might bind cooperatively to EIF4G. As this shortens this to about 400 amino acids now, so it's much more deliverable. We also checked the SCN1A mRNA expression in this experiment, compared both to a non-target guide as well as our effector negative control, where GFP is the effector with the on-target guide. And we see that there's no change in mRNA level, showing that this is in fact activation rather than mRNA stability or RNA binding site or something like that. So I'm going to skip some steps just to get to the important part, is that we validated that we could also do this in vivo. So we used the Dravet syndrome mouse model and delivered an AAV encoding the SERTs and protein and the guide to neonatal mice. So like I mentioned before, is a childhood epilepsy, so the mice typically start showing symptoms around P21. So our first experiment was to dose the mice early in life at P1 and then check that we were changing protein expression before they showed symptoms at P20. So we extracted protein tissue from the hippocampus and cerebral cortex, and then probed NAV1.1 levels by Western blot. So this first lane is a healthy wild-type mouse, so this is healthy protein expression. And then after that, we were alternating haploinsufficient mice, either treated with vehicle or treated with the SERTs AAV if it has a plus sign. And we also probed for SERTs expression in the brain by that middle blot there. And we could see when we normalized that we had about 30% increase in protein expression in the brain, which obviously is not full rescue, but previous studies in Dravet syndrome mice have shown that even 20% can lead to phenotypic improvements, so we were motivated to continue with the study. And we also showed by RT-qPCR that in vivo we're also not changing mRNA levels. So into the phenotypic study. So like I mentioned before, particularly 21, and very common is just about 50 mice sufficient for SC1A will die from death in epilepsy. We did see that female mice, where mice treated had about 50% survival, but mice treated with TV had about 87% ability. Interesting is in the some reported. So we did a separate mice where we seizure susceptibility to temperature. So this a addition in that many children or parents of Dravet syndrome often diagnose when their child has and that fever causes seizure. So you measure mouse's internal body temperature to raise the ambient temperature to record the temperature that they have a seizure. And we found that mice treated with vehicle onset of seizures around 40 C, while mice around 41.5 C, with 2 mice in this group not recording seizures at all. So we're able to see minor but significantly, statistically significant phenotypic improvements, which really help bolster our confidence with moving forward with this strategy. We've also expanded this to other targets, some other chromatin remodeling enzymes, and such as CHD2, where we repeated a similar pipeline to find guides that can upregulate CHD2 expression in N2A cells as well. And we've also done this with ARID1B, showing that this is generalizable. So I'll end my talk by giving some very preliminary data with our targeting on EHMT1. We designed a similar reporter for EHMT1, as I mentioned before, using 3' UTR sequence information that we gathered from long-read transcriptomic data gathered by the Casfree lab from iPSC-derived neurons. This is a kind of a, I mean, only RNA people would really care about this, but in the NCBI database, there's like a lot of different 3' UTRs for EHMT1 that are annotated. But in this iPSC-derived neuron line, there's only one sequence that actually associates with full-length EHMT1. And it's isoform 1 on the NCBI database if you're interested. But so we've completed this, and the 3' UTR EHMT1 is relatively short compared to a lot of other neurological transcripts. So we were able to completely tile with about 11 guides, where we found guide 3, but really up into guide 3, we saw some activity by this assay. But as we move into N2A cells, so far the activation has been pretty minimal. We've found reproducible but minimal activation with guide 3 so far, but this is still optimization. So particularly what we're trying to do is we're trying to also move neuron-like cells. The N2A cells seem to work well for getting a baseline if things are working, but can be hard to push over like a 50% threshold of activation. So we're working around this, with lentiviral delivery to SHSY5Y, with and without differentiation, but I'm open to other people's suggestions on good cell lines to kind of do these assays with around the human guide sequences. And then also we're working on multiplexing certs. So here we're using multiple guides that show some activity to see if we can bind multiple certs to one mRNA and potentially increase translation. But just to summarize, we developed certs4GT3. It's a programmable tool to increase translation from disease-relevant transcripts. It's minimal 600 amino acids, so flexible packaging is very possible in AAV vectors. But a limitation to RNA targeting is mutations can't be dominant, negative, or X-linked, so that's an important caveat. So future directions, obviously improving efficacy is our biggest priority, either by further engineering the platform or its deployment, and then also answer outstanding questions on specificity, immunogenicity, and generalizability. So I'd like to thank my group at the University of Chicago, my boss, Brian Dickinson, for helping oversee this work, and of course, funding sources, including IDefine. I'm really excited to be part of this research community and excited to stick around for the rest of the conference into the family section to learn more about all the cool work that's being done and the people doing it. So thank you.